Okay. Okay. Hey Nancy, how are you?
Good, how are you?
I'm good. I'm gonna let you know I'm recording today's session. Then I'm gonna upload it as a YouTube video on Discord. Okay.
Okay.
And I'm wearing these just because the microphone is over. There's a mic in here, so they can hear me better in the video. Okay. But
Okay.
I don't have um, music playing. I can hear you guys. Okay.
Awesome.
So I am gonna try to dedicate more time. I'm free, so we're definitely gonna go an hour, maybe an hour and a half, okay?
Okay.
And then I'm just gonna get started. Essentially, we pretty much went through the whole material last session with you, Samantha, and then with you, Nancy, also. Like we kind of pretty much went over it, and then it's my understanding that last lecture he just went into more depth in the cell cycle, right? And that's where we pretty much stopped.
Yeah.
Okay. Okay. So, going back all the way to the beginning, we started with like hypotheses and how to make like scientific reasoning, pretty much, and thinking. So, main things to remember: independent variable and dependent variable, right? So independent variable is the one that we're able to manipulate our own will, independent, and then dependent is the one that it depends on the independent variable. So dependent variable is usually what we measure. So for example, if we're trying to see different temperatures at different altitudes, we can manipulate which altitude we're choosing to take the temperature easily, just our free will. So that's independent. And then temperature depends on how high we are. So that's going to be dependent variable. So usually on a graph, you're going to see on the x-axis, you're going to see the independent variable, and then on the y-axis is going to be dependent. Usually, not always, but most likely. Then when Dr. E is gonna ask you to make a hypothesis during this test at the end, you're still gonna use that if, comma, then. So if, the example with the altitude, if we go higher above the sea level, then there's gonna be a decrease in the temperature of the surrounding atmosphere. Okay, so it's that if that. With that, you can't go wrong. And then, last thing I'll say about this is, most likely your answer about this is gonna have to do like in this question on the exam, because there will be a question about this on the exam. The open response, you know for sure that that's gonna be on the exam, so might as well at least be prepared for that one, right? So most likely if it's gene editing, you're gonna have something to do with CRISPR-Cas9, right? So if there's a mutation, then you can always use this technology to fix a mutation in your germline to fix a disease. And then obviously when you are creating your experiment, make sure you have a control, yeah? So when you're testing whatever your hypothesis, you're gonna write out, there's also gonna be a control that's not gonna have whatever you're doing. That makes sense? Alright, so that's pretty much that chapter, yeah, like the first lecture. So second lecture is going to be cell types and evolution. So essentially prokaryotes versus eukaryotes, we know that. prokaryotes, no nucleus, no organelles, not really an endomembrane system. They still have a cell membrane, and they might also have a cell wall. The cell walls in prokaryotes are going to be made up of peptidoglycan or lipopolysaccharides, if I'm correct. And then gram-positive bacteria is the one that has a higher concentration of peptidoglycans. Gram-negative bacteria is the one that has a lower concentration of peptidoglycans. Gram-positive, negative. Cell wall in eukaryotes, plants have cell wall. Mammals don't have cell walls, but like some eukaryotes also have cell walls. So it's not only prokaryotes for a cell wall. And then so mitochondria and chloroplasts, they were evolved through endosymbiosis. Remember that? I don't know. Might come up. So this was the event where a prokaryote ate another prokaryote but did not digest it. Instead, they started a symbiotic relationship that that created rise to like organs. So endosymbiosis is essentially what created eukaryotes. And then the term LUCA, L-U-C-A, is going to be last universal common ancestor. So this is like if you go back far enough, this is like an idea essentially that all of us came from one bacteria, right? I can't really come up with a question about this, but I feel like on our final exam we did have something about this. Just remember that LUCA, it should be a simple question as long as you remember what LUCA is. Okay, that makes sense, right? That should be easy. So biochemistry, we have two types of bonds. Covalent and non-covalent bonds. No, okay, three types. Covalent, non-covalent, ionic. So ionic is gonna be metal and non-metal, right? In the body, they're strong bonds, but they're easily dissociated in a liquid environment. So in our body, we're not gonna really have ionic bonds because as soon as you put an ionic thing, like for example salt, NaCl, it dissociates into ions. Okay, so in the body, ionic bonds are not really present. Then covalent bonds, we're sharing electrons. It's non-metal and non-metal, and there's gonna be lots of those. So H2O, the bond between oxygen and hydrogen, it is covalent. For example, bonds between amino acids in a peptide, they are covalent. There's actually a bond, electrons are actually shared.
Non-covalent?
Covalent, yeah. So these are strong bonds. It requires a lot of energy to break them. Usually, to break a bond like that, you need ATP, or to create a bond like that, you also need ATP. So then, non-covalent bonds are the bonds that are you don't really share electrons. Okay, so it is charges that like play a role in it, but you do not form an actual bond between two things. So three types of them are gonna be van der Waals forces or London's forces. Then there's gonna be hydrogen bonds. Is there more? Or is it just two?
London dispersion.
So that's van der Waals, yeah. I guess, yeah, that's the two main ones. And hydrophobic interactions we can also see. But mostly we're going to focus on hydrogen bonds and the van der Waals forces. So hydrogen bonds are the stronger ones. Out of non-covalent, hydrogen bonds are the strongest one. So hydrogen bond is the strongest non-covalent bond. But covalent bonds, they're obviously stronger than any non-covalent bond because electrons of two different atoms are actually interacting. Like we're sharing them. So overall, covalent are stronger than non-covalent. But if you have a question that's asking what's the strongest non-covalent bond specifically, it is going to be hydrogen bonds. So now, just I want to make this really clear. So for example, hydrogen bonds, they're used in DNA, double helix, to maintain the structure. So that's the nucleotide base pairs, they're bonded through hydrogen bonds. And then, so I said that covalent, so in water, this is a covalent bond because hydrogen and oxygen, they're sharing electrons. This has 6 electrons originally before they connect. This has 1 electron, this has 1 electron. And then together they have, since they are sharing, this is gonna be 2 electrons, this is gonna be 2 electrons, this is gonna be 8 electrons, and that's why they're all mutual, because they're sharing. But then oxygen has a much stronger pull on those shared electrons. So the electrons are gonna be closer to oxygen, like physically, they're just gonna be, instead of going like this, where they're shared equally, it's just gonna be like this, you know? So the electrons are gonna be closer to oxygen. Since the negative particles that are meant to be shared are closer to oxygen, oxygen is gonna get a partially delta negative charge, and hydrogens are gonna have a partially positive charge. And so this partial thing is gonna make it possible for a non-covalent interaction to occur. So meaning it's partial, it's not even a full charge. So they're not gonna form a full bond, but since this is partially positive, this is partially negative, and positive and negative attract, there's gonna be that sort of attraction right here. So water is able to participate in both covalent and non-covalent hydrogen bond interaction. So meaning they're not mutually exclusive for any molecule. And so, like, there might be questions about what makes water such a great solvent. So it is that whole characteristic of it being partially charged, and also it creates high heat capacity, meaning that it requires a lot of heat for it to boil, which is also good for living systems because it's a stable solvent. It also creates, you know, that like flowy movement of the water that you're able. It's not like it's not random. It's just very smooth, and all that is because of hydrogen bonds. And then just And then just a quick review, 4 main macromolecules in our body. It's gonna be protein, lipids, carbohydrates, and the last one is gonna be nucleic acids. This is essentially what we are made up of. Does that make sense?
Are you gonna color the tap?
The what?
The tap on the marker.
No. So proteins and folding. I remember we were like struggling a little bit on this when we were going over it originally. So there's 4 structures: primary, secondary, tertiary, quaternary. Okay? And so every single protein is gonna have at least up to tertiary. Some are gonna have all of them. And it goes in a progressive manner, like in the complexity, progression of complexity. So primary is going to be the easiest. It's just the sequence of amino acids. So if it's V, like valine, alanine, I don't know, whatever you want. So when I'm just saying the amino acids, I'm describing the primary structure. Okay. So that's what's made in the ribosome. That's what's coded by mRNA. And then everything that you need for everything else is in that mRNA. So you don't need anything else. All the information for secondary, tertiary, and quaternary structure, it's already in the sequence. So then once it's made, it just folds into it. So secondary structure. I'm gonna write it here. So primary is gonna be sequence. Secondary specifically describes.
Russ, there's a glare on the top left corner, so maybe just write it a little bit lower.
Right here is good.
Like,
Yeah, it's not.
yeah, I'm also kind of blind, so I'm just not being able to see.
Thank you.
Yeah, that's good. That's good.
So secondary is gonna describe specifically alpha helix or beta sheet. So alpha helix is literally going to be a helix, and then the amino acids are going to be linked along here, and then the functional groups like stick outward, I believe. So it's like it just takes like this spiral thing form, and then a sheet is going to be like multiple of them aligned like this, which creates a sheet. This, I don't think this is important. Just remember, secondary is going to be alpha helix and beta sheet. It cannot be anything else. It cannot be anything else. So it's just one or the other. Then tertiary structure is going to describe the way protein folds in on itself. So when a protein is synthesized, it's going to have hydrophobic areas that doesn't like water. It's going to have hydrophilic areas that does like water. And so depending on that patterns, you're going to want hydrophobic areas to be inside of the protein, protected from the water outside, because that lowers the energy, and that's good. The protein is more stable. And then hydrophilic areas are going to want to be on the outside, because they want to be interacting with water, which once again lowers the energy of the protein, which is good because we want it to be stable. So it's folding on itself. And then quaternary structure describes proteins that have two separate subunits coming together. So for example, if one subunit is synthesized from one mRNA and the other subunit is synthesized from another RNA, and then they come together, and only after they come together they become functional, you would say that the quaternary structure of this protein is made out of two subunits. That's just what it describes. And they can be different, they can also be the same. You can have the same subunit, you just need 2 or 4. For example, in hemoglobin, we have 2 beta, 2 gamma, right? Or 2 beta, 2 alpha, like subunits or whatever, like depending if you are like in the fetus or a live person. So its quaternary structure is made up of 4 subunits and of 2 pairs of different subunits. So that's what it describes. So like more than one subunit.
Can you draw that out?
So it's more than one subunit. It just describes if there's more than one subunit, then you are gonna have a quaternary structure.
How does it look like?
Like
Quaternary.
hemoglobin, for example. Obviously, this is my artistic rendition, but it's gonna be like this thing right here, and then this one is gonna be the same, and then this right here, and then this right here, and like this is one functional protein. This is the same, and this is the same.
So if you have a three subunits, would that cause issues, or it would be
In
fine?
hemoglobin specifically?
Like, or yeah. I
In
guess
hemoglobin specifically, yeah. You need like this is it's only fully functional when it's this. I think when one of these subunits is non-functioning, that's how sickle cell anemia happens.
Okay.
But I think in that case you're missing 2 of them, if I'm not wrong.
But would he ask that possibly on?
About sickle cell anemia specifically, or what?
Or like if one of the subunits was like not.
No, I mean I feel that's a fair answer. You should be able to answer that. If one of the subunits is not working, then most likely it's a function is going to be affected to some degree.
Okay.
It might be able, depending on the case, it might be able to like compensate for it somehow, but the function is going to be affected.
Would it be the same if there's a subunit that's not functioning as if there was just a missing subunit?
If it's if it's not properly folded, then yeah.
Okay.
But I mean, it could provide some stability.
Mhm.
What is the molecule that helps with misfolded?
So chaperones, like chaperones.
Yes.
Like so, think like chaperone, someone who follows you to make sure that you get to your destination, right?
Okay.
So this is exactly what it does. Once you have been synthesized, like I said, all of the information that's required is already in the sequence. So there's no additional information going in there necessarily, but sometimes there are chaperones and they can do 2 things. Like sometimes chaperones start right off the bat, and so it's gonna fold in on itself and it's gonna fold in into the easiest conformation that it can if there's no assistance. But sometimes maybe for a protein to be fully functional, the functional conformation is not really the easiest one, but it's also like one of them, right? So a chaperone is just gonna guide like a little bit. It's gonna nudge it towards the right direction, so it doesn't fold into the one that we don't want. But they also can refold proteins through, if you fold it incorrectly for some whatever reason, they can like unwind them and give them a second chance, or they can also like again nudge them into the correct direction. So chaperones are the ones that like gonna help with protein folding. And then protein misfolding is like neurodegeneration is mostly due to protein misfolding. Just remember that as a clinical example, okay?
Like Alzheimer's disease.
Exactly, yes. So if it's like if there's a question about asking you about clinical treatment of somebody with Alzheimer's disease, and if it is a genetic issue, CRISPR-Cas9. If it is something else, not genetic, chaperones. I think that should be cover you. Okay. So for chromatin, there's gonna be like a nomenclature. We went over it, but it's just important that you remember it. Okay. So chromatin in general just describes chromosomes when they are in the interphase. Does that make sense? So when you are not in mitosis, you don't have chromosomes. You have chromatin, which is just like a little soup of everything. And then chromatin itself can be further subdivided into two types. So we have euchromatin and we're gonna have heterochromatin. So heterochromatin is the one that's more condensed. It's harder to transcribe. And euchromatin is gonna be the one that's less condensed, that's easier to transcribe. Transcription to translation. Yeah. So I don't know whatever like mnemonic device or memorization technique you want to use, but just make sure that you're clear on that chromatin is like the general thing, general description. Euchromatin and heterochromatin are more descriptive of chromatin, and they're kind of opposite, working against each other. And in a nucleus, you can have over here in this area, you have euchromatin, for example, and then over here in this area, you have heterochromatin, like depending on which genes you need to express. And so then we have histones. Histones are proteins with a DNA wrapped around, and so modification of histones is going to control the balance between these two. And acetylation, so the addition of acetyl is going to make more euchromatin, and then methylation is going to make more heterochromatin. Okay? Now, this is when we're adding. It's also important to understand that if we are demethylating, then we are making more euchromatin. And then if we are deacetylating, we're making more heterochromatin. So opposites, right? Addition of acetyl makes euchromatin. Removal of acetyl makes heterochromatin. Addition of methyl makes heterochromatin. Removal of methyl makes euchromatin. Okay. And so DNA replication happens in the 5' to 3' direction, and there's gonna be the leading and lagging strand. Okay, I'm gonna make a sketch of the replication fork here. I'm gonna erase it later because I need the space, but let me know. Okay. So when we're moving 5 to 3, this is the leading strand. Over here It's the enzyme helicase unwinds. Helicase removes the two things from each other, and then topoisomerase. Does anybody remember which one like prevents it from supercoiling? Is it topoisomerase?
That sounds right, I think.
Sounds right, right?
It sounds good.
Yeah,
Alright.
I think so.
I'm pretty sure you are right.
Yeah. So the topoisomerase is the one that prevents the supercoiling of the DNA. The replication fork is made up of obviously like a lot of enzymes, but I think those are the two main ones that we were talking about when we were actually learning about this during the lecture. So we'll keep it at that. Then there's DNA polymerase. Okay, DNA polymerase is the one that actually synthesizes the DNA. And then there's like alpha, beta, delta. In reality, they all do different things, but we're not gonna worry about that. But then DNA synthesis can never start by itself. DNA synthesis always needs a primer, an RNA primer. RNA primase is gonna initially come in, make a primer right here. Then DNA polymerase is gonna bind to this RNA primer, and it's gonna start making DNA. Okay, so on the leading strand, it binds once and it starts going until it's done. Later, after it's done, I can't remember, but like one of the other DNA polymerase, alpha, beta, delta, whatever, like they remove RNA, they put their own DNA there, and then ligase, a separate enzyme, is gonna connect. Okay, so I'm gonna actually draw. So this is removed, right? And then instead of it, we're placing this, but then there's gonna be a tiny hole right here because we synthesized them at different times, and ligase is gonna connect the hole. Okay? This is the leading strand, easy. Then we have the lagging strand, and it's lagging because we can only move in 5 to 3, so we have to move from the point of unwinding to the other direction, and then we start here, go move this way, but then as we're moving this way, there's more and more DNA being unwound here, so we're gonna have to constantly jump back and start over and over again. And so essentially, I just want you guys to remember the word Okazaki fragments. So Okazaki fragments describes this, like the fragments that are created because we constantly have to start over and over again because a new 5' end is being unwound. So lagging strand is lagging because it has Okazaki fragments, and everything the same is going on over there with the ligase, with the primaries, with the DNA polymerase. It's just you have to start over multiple times. So helicase, topoisomerase, DNA polymerase, primase, ligase, Okazaki fragments, and then 5' to 3'. So RNA generation, central dogma, if he asks, it's DNA to RNA to protein, like just that's like definition, right? So transcription of coding RNA happens by RNA polymerase 2. And so RNA polymerase 2 binds to the promoter region on the genome and it synthesizes mRNA. And then once mRNA is synthesized, it contains exons and introns. Introns are like intruders. Don't think like it begins with in, which might make you think that you're gonna keep it, it's gonna stay inside, but no. Introns, think of it as intruders. Exons, I don't know, think they're gonna exit the nucleus, like they're actually gonna exit the nucleus, they're not just gonna get degraded. So we're keeping exons, cutting away introns. First thing, we did that. Second thing that we need to do is we need to have a 5' methyl G cap. So if I'm saying 5', we are gonna start, that's gonna be at the head, and it's gonna be just a modification of mRNA at the top. And then we're gonna have a 3' poly A tail. So and 3'. So this is gonna create, this is what binds to ribosome, okay? So without this, you can really bind the ribosome. So this is like a primer, right? Think of it as a primer for the protein to actually be able to be translated. And then poly A tail, I think of it as a telomere for an mRNA. It's a whole bunch of A bases at the end of mRNA. So in case we do begin degrading, we're losing just these like useless adenosine bases that we don't really care about instead of actual coding material. And after this, we are a mature mRNA. So maturation of mRNA, 3 phases. Exons, introns, 5' methyl G cap, 3' poly A tail. And then, just also like some simple stuff. Ribosome binds to mRNA. tRNAs bring amino acids based on which codon we are reading. So, that's that. Codons, this might come up, I don't know. But so, so for codons, start is always going to be AUG. So think fall starts in August. Like your fall semester starts in August, so AUG. And then your three stop codons are going to be UAA, UGA, UAG. If I don't know, I don't want to say you're not going to need this. I'm pretty sure you won't need this, but like I've said this before, and then I've seen questions that I said you're not gonna need this again. So if you want to remember this, you can remember this as you are annoying, you go away, you are gross. Okay?
UGA what?
You are gross. You go away, you are annoying. Like, you know, you want to, like, it's stop codon, so like, you know, you're saying negative
These
things
are
for
all
it to stop.
stop?
Yeah, so this is gonna stop translation. I'm gonna get some water, I'll be back. Gram positive or gram negative?
Oh, gram.
Sorry.
My goodness, right, grant.
So it's peptidoglycan concentration in the cell wall. So for membranes and transport, in general, our cellular membranes, they are described as having a fluid mosaic model. We know that, right? Just like that it moves. And then, so we have unsaturated fatty acids, and we have saturated fatty acids. So saturation of a fatty acid describes presence of hydrogens on that fatty acid. Our fatty acids are going to be just a lot of carbons linked together, and then every carbon needs to have 4 single bonds. Okay, if it's single bonds, it needs to have four bonds every carbon for it to be stable. Otherwise, it's going to have a charge. It's going to be unstable. So right here, this carbon, it has 1 bond to a carbon, another bond to a carbon, and then the other 2 are 2 hydrogens. And you guys know that in OChem, right? Just for you haven't taken OChem if I remember correctly.
Fell twice.
You did what?
I fell twice.
Oh, well, I mean, it's okay. We remember this though, right? Just hydrogens. And so, saturation, so we have 2 hydrogens on every carbon except for the last one. The last one's gonna have 3, right? Because there's no carbon there. So if we are unsaturated, means that we don't have
We're missing.
2, we're missing some. We're missing 2 hydrogens most likely. So then we're losing 2, double bond is formed. Okay, so if you see a double bond on an amino acid, it means that it is unsaturated. So just as long as you remember that saturation describes the presence of hydrogen, you'll be fine. Then unsaturated amino acids are like good, red, or whatever, and then saturated are less good. So saturated amino acids, they're gonna be like very straight, right? Because there's no double bonds. Unsaturated amino acids, double bond creates a cloud of electrons in that place, which bends it in a certain direction.
Unsaturated?
Unsaturated, yes, unsaturated. So double bond, if double bond is present, you are unsaturated. You had to have lost some hydrogens for the double bond to be formed. And so this movement of the fatty acid is just gonna make it take up more space, which is gonna increase the fluidity of the cellular membrane. So unsaturated fatty acids increase fluidity of the cellular membrane. And then saturated fatty acids, they're all straight, they all have that same conformation, so you can really pack them really close to each other, which is gonna make it hard for them to move. So saturated fatty acids are gonna decrease cellular membrane fluidity. So that's for that. Then cholesterol is a whole separate thing. Cholesterol, it's like a lipid. It's a lipid. It is derived from do you guys remember this thing in organic chemistry? What is it called? Like it's like this.
A python?
No,
What is that?
isopropene.
What?
Isopropene. Is it? I think it is.
Oh, like that whole
Isopropene.
This thing right
I
here.
think it's isopropene. Let's double check.
But how
Propyl-
does your name mean?
No, it can be propyl because propyl is 3 carbons. 3, 4, it's 4 carbons. MIP-B, it's butyl.
Isobutene.
Okay, you know what? This is actually I'm getting us off track. I don't know why I wanted to talk about this, but
Shouldn't it be isobutene because it has a double bond and
What's this?
Oh. I almost-
and it has a methyl group on the middle carbon.
oh, sterol. Sterol. Isn't that what it is?
3-methylbutene?
Oh no.
Oh no, sterol is dye.
Is it ethylene?
That's not ethylene because ethylene should be two carbons. That has 5 carbons. If you include the methyl group
Yeah,
to the
that's right.
butene, it should be pentene. Isopentene.
Anyways, sorry. I'm so sorry. But I wanted to remember the word sterol. So sterols are gonna be this thing, these. So 1, 2, 3, 6 member rings in this arrangement, like next to each other, diagonal up, and then one again next to each other. So this is a sterol, and cholesterol and all steroid, which we're gonna talk about later about signaling. They all start with this, and then this is modified. Okay. So cholesterol is gonna act in membranes as a buffer, and which means that when temperature increases, our fluidity also increases because there's more energy for them. There's more energy available to use for movement because we're hot. So at that point, cholesterol is gonna act to hold everything together. So if we're increasing temperature past our normal homeostasis, cholesterol is gonna function as a saturated fatty acid. It's gonna decrease fluidity to make sure that we don't get so hot that the membrane rips. And if it gets too cold, there's less energy available. If there's less energy available, we begin to slow down and we're losing some of the fluid membrane mosaic properties that allow for dynamic exchange of nutrients from the outside to the inside. So if it gets too cold, things are gonna stop and we're not gonna be able to transport certain things. So in those points, cholesterol is gonna act as an unsaturated fatty acid and it's gonna upregulate the fluidity of a membrane. So buffer. So passive versus active transport. Passive transport, energy is not required, usually from high concentration to low concentration, diffusion. Active transport, energy is required for whatever reason. Maybe the molecule is too large to move through by itself, maybe we're trying to create a gradient, or so we're using ATP. And then there can also be facilitated passive transport. Okay, so passive transport, you would move directly through the cellular membrane. Facilitated passive transport, you would not require energy, but you would require some sort of channel, a protein channel for things to move. Most likely it's gonna be an ion. Ions are charged. Charged things cannot move through the cellular membrane because it is a non-polar, so it doesn't like charged things. It's gonna repel them. And if you do want a charged particle to move through, you need some sort of a entranceway, which is a channel, but no energy required. And then facilitated active transport is like the sodium glucose transports. So when you are using when you're using the high to low concentration of sodium to move glucose in as well. Remember that? And then regular active transport is gonna be like the sodium potassium pump in the neurons that creates the membrane potential. So you're using ATP to pump things, and then so if you remember sodium potassium ATPase, that's what it's called. Then who remembers what goes inside, what goes outside, and how many?
Out for sodium and then
Yes, out for sodium.
in for potassium.
In how many? What? 3 sodium, 2 potassium. Yes. So if you don't remember, think Na like no. You wanna get it out. So sodium moves outside. K like okay, you move it inside. There's 1 letter here, 2 letters here. 2 letters, 3 goes out, 1 letter, 2 goes in. Okay. So for the membrane potential, like I said, sodium potassium is what mostly generated. They're both positive, but like we just found out, since it's 3 sodium that's leaving and 2 potassium that's coming in, we are less positive on the inside than we are outside. So being less positive is also similar to being negative. So we can arbitrarily say that the inside is negative compared to the outside, but they're both positive, okay? So just don't get confused. So in the resting membrane potential, we have positive outside, negative inside. So then in the synapse, we releasing neurotransmitter binds to receptor. Once that happens, we are opening ligand-gated ion channels. So this is a ligand, and it opens the first in a couple of initial ion
Is that for
channels.
8 or 7?
8. Membrane potential. Sorry, Nancy, you can see. Is that better? Can you see this right here, what I'm showing?
Yeah, that's good. Thank you.
So ligand binds to receptor. It opens initially the ligand-gated ion channels, which open, and then there's a high concentration of sodium outside and a high concentration of potassium inside. So they're gonna automatically flow in opposite direction, and that's gonna create a charge locally around those channels. It's not gonna be a lot, it's very minimal. But that local change, so right here we have the first channel that's ligand gated, and then it's gonna open the channel that's next to it, which is voltage gated. And so without binding, it just, the local change in voltage is gonna activate this, and this channel is gonna do the same thing. It's gonna locally change right here, which is gonna activate this channel, and this is how the membrane potential travels across the neuron, all the way to the end. Okay, so it is all or nothing. If you are able to reach that voltage requirement, we have a fire. If you're not able to reach, we have no fire. So neurons are all or nothing. It's like binary. It's 0 or 1. It cannot be 0.5. But we are able to regulate intensity of our neuronal signals, like I'm sure everybody knows. That happens through frequency of firing. So this is a graph, and then this is the x is time and the y is the voltage inside. Like if we have something like this, then we are firing a lot. We're feeling, for example, a lot of pain or whatever. It's like something very hot. But if we are firing instead of maybe every 10 seconds, oh no, every 1 second we had like 3 neuronal fires, then it is a less severe response to a stimulus. Okay, so
All or nothing.
Do you think you would need to know the certain like pain receptors? Because he talked about it like in class, and then also for our homework we've done like.
The only thing I would want you to remember is that nociceptors. That's the receptors responsible for pain. Nociceptors.
No season.
So for neurons, you have these nociceptors. Pretty sure. Yeah. So this is a type of sensory neurons, and then, but in general, you have motor neurons. You have sensory neurons. Motor neurons are the ones that like allow you to move, and then sensory neurons are the ones that like allow you to feel sensations.
What are these?
Huh?
What are these?
Sensory. So this is for pain. This is a category of pain receptor or of sensory receptors. Pain receptors. I mean, also there's like the myelin sheath. Yes. So Schwann cells. Okay. So if this is the axon. Okay. So just this is really quick. Myelin sheath, they are these things. They're made out of lipids and they're like insulators. So when we are doing that thing, when we are locally changing charge, it's obviously like it's done in less than a second for us, but on this level, it takes some time. So the body, in order to increase the speed at which signals travel, it makes these insulators. So instead of having to travel from here to here, you travel from here to here, jump over, from here to here, jump over, from here to here. Okay? And so those are called myelin sheath. They increase the speed of your neuronal signal fire. Okay, Nancy, I want to move you to the other board. Are you done copying everything that you're seeing right now?
Yeah, I'm good. Thank
Do
you.
you need to see anything else before we move on?
No, we're good.
Up? Oh, right. I'm gonna start right here. Right.
There you go.
Okay. So, I don't know. For metabolism, I'm gonna go over it really quickly, but then there's like so much detail. If you have any specific questions, let me know, okay? But so, overall, metabolism is glycolysis into TCA the citric acid cycle, and then we're gonna go into electron transfer chain. When we are in the glycolysis, that happens in the cytoplasm, and then TCA happens already in the matrix of mitochondria, and ETC, the proteins of the electron transfer chain, they are embedded into the inner membrane of the mitochondria. So we have glycolysis, cytoplasm, into TCA, which is the citric acid cycle, which is matrix of the mitochondria, into ETC, which is electron transfer chain, which is embedded into inner membrane of the mitochondria. So in glycolysis, we're gonna take one molecule of glucose and we're gonna break it up into, first we're gonna, at the end of glycolysis, we form two pyruvates. Then those two pyruvates become two acetyl-CoAs, and each acetyl-CoA enters the TCA by itself. So, essentially, I just make sure you understand that one molecule of glucose entering glycolysis is gonna start two cycles of the TCA. Does that make sense? Because one glucose makes two acetyl-CoAs, and then all of that is gonna go to ETC.
2, 1.
One glucose makes two acetyl-CoAs. Acetyl dash CoA. Coenzyme A. Acetyl coenzyme A.
And that makes the
So one glucose, two acetyl-CoAs. Essentially, we are trying to make NADHs and FADH to send those to the ETC, because ETC is the biggest payoff, right? Glycolysis gives us a little bit of ATP, this gives us GTP, and ETC gives us most of our ATP. NADHs go to complex number 1, FADH2 go to complex number 2. That's why NADH yields approximately 2.5 ATP per molecule, and FADH2 yields approximately 1.5 ATPs per molecule.
about 3 ATPs for everything.
I'm sorry?
About 3 ATPs for everyone.
No, you need to remember these numbers, okay? So NADH is gonna be 2.5 and FADH is gonna be 1.5. And it's not actually half, you can't really have a half ATP, but this just means that half the time you make 2, half the time you make 3. So on average, you make 2.5. Because it's not a perfectly efficient process. Sometimes there's mistakes and you make 2. But sometimes you do good and you make 3. So then there's fermentation, anaerobic fermentation, which occurs when there is no oxygen to act as a final acceptor of electrons in the ETC. So we are overly relying on glycolysis to generate our ATP. And so essentially, what you need to remember is that you start working out, right, or whatever, your energy starts producing energy. It goes glucose, goes ETC, no, goes TCA, goes ETC. but then all of a sudden it realizes that, oh, there's not really a lot of oxygen, and then you quickly run out of oxygen. So you keep doing you keep doing glycolysis, but glycolysis generates it needs NAD pluses to generate NADHs. Without NAD plus, you can't move forward. So at some point you use up all of your NAD pluses. If there's no NAD pluses, you can't even do glycolysis. So you're trying to regenerate NAD pluses. So instead of sending NADHs to ETC and actually getting ATP, all you're doing is just breaking off this hydrogen and wasting NADH, essentially just to get at least a little bit ATP from glycolysis, because there's no oxygen to accept the electrons from NADH. And so that generates lactic acid in us humans as a byproduct. So anaerobic fermentation generates lactic acid, and in bacteria and in yeast, it can also generate alcohol. And then the last thing I'll mention is the Warburg effect. So Warburg, W-A-R-B-U-R-G effect. And this is what happens in cancer. And this is cancer running glycolysis more preferentially compared to TCA, because glycolysis is sort of a quicker, like a faster energy source. So you're able to get less energy but quicker, so that's what fuels that rapid growth. And then you're also able to use all of the intermediates in the TCA as building blocks. So instead of using that to build energy, you're using it to generate more and more structure, which is what cancer wants to do. Okay, I'm gonna move on from metabolism. Does anybody have any questions? This is like part one of the new materials to my understanding.
The Warburg effect is also beneficial for cancer cells because there's less, like since it's in the cytoplasm, there's less ways for it to get tagged for cell death, right?
Yes, because if you don't involve mitochondria, there's less chances for you to go through apoptosis. Okay, so I'm gonna lower the screen and see, do you need more time?
No, I'm good, thank you.
Okay. So for cell signaling, I'm a little tired of writing, I'm not gonna write anymore. So you have protein signals and you have steroid signals, like lipid signals. So proteins are going to bind to cell receptors on the surface of the cell. Think insulin binding to insulin receptors, and then steroid and lipid-based hormones. They're gonna because they are non-polar. Remember, like we're connecting back to, since they are non-polar, since they look like sterols, they're able to easily move through the cell membrane, and then they usually bind to a protein inside and act as transcription factors inside of the nucleus. So there's RTCase as a receptor tyrosine kinases, which is when it's like two dimers that when they bind the same ligand, they come together, and then on the inside they phosphorylate. And that cell sends a signaling cascade inside of the cell. And then we also have GPCRs, which is a G protein coupled receptor, which is a receptor that has
It has like 7 intermembrane domains. 1, 2, 3, 4, 5, 6, 7.
GPCR.
GPCR, G protein coupled receptor. And then it has the alpha, beta, gamma, 3 subunits. That when we are in the inactive form, we have the GDP bound to the alpha subunit. Once the ligand binds on the outside, GTP is switched out for GTP triphosphate, so that makes it active. Beta and gamma subunits leave the alpha subunit, and then alpha subunit activates whatever is next. Okay, I think I remember the number 7, please. And remember that it's alpha, beta, gamma subunits. Remember that GTP switches for GTP on the alpha subunit. The alpha subunit is actually important now.
You said GDP to GTP?
Yeah,
Or-
a GD, like die, like 2. When it's D, it's inactive. And then when it's T as in try, we're active. And then, so cell cycle is regulated through interaction of cyclin-dependent kinases and cyclins. Okay, so cyclin-dependent kinases, they are always present, and then cyclins is what is synthesized, and then cyclin binds to cyclin-dependent kinases, which activates it, and then those CDKs are able to do their thing. So me and Nancy came up with, I think it was like 64211. Nancy, what was it? Do you remember?
It was 4211.
421, and then one with the letters?
D4A2A1E1.
So this is gonna make sense right now. So this is like, remember he said he was gonna ask you guys, he's not gonna give you like a single one, but he's gonna give a pair, right? If he's gonna ask anything. So this is the cell cycle. We're always gonna start at G1. So this is gonna be G1, this is gonna be S, this is gonna be G2, and this is gonna be mitosis, okay? So in G1, we have cyclin 4, and no, it's CDK4, right? CDK4 and cyclin D, right?
I think so. I think so.
The cyclins have the letter and CDKs have the number.
Correct.
Correct?
Yeah.
Okay. Yeah. So the cyclins have the letter, the CDKs have the number. So
I thought we said
Nancy, is the last one also? You know,
Oh,
yeah.
that one's B.
Oh, so it's DAAB.
D-A-A.
Okay, so it's DAB, yeah? DAAB.
A-B.
And then 4211. 4211.
Yes.
4211. So in G1, you're gonna have CDK4 and cyclin D bound together. In S phase, you're gonna have CDK2 and cyclin A bound together. In G2, you're gonna have CDK1 and cyclin A. In M, mitosis, you're gonna have CDK1 and cyclin B. So if you have a question like this,
I thought we don't remember this because also don't cyclins go throughout multiple phases?
yeah,
Might be
so that's why you have A here and A here, but it bounds to different CDKs. So they do go through different phases,
No, I got you,
but like the pairing is, so that's what I thought, that you have to remember the pairs, not every single individual one, but the pairs.
I think he said that we just need to know what they do. I don't think that we need to remember when they have the pairs either. I don't know, just know it just in case, I guess, but like, I don't think we need.
This is easy to remember, okay? This
Yeah.
is easy to remember.
Okay.
Like 4, 2, 1.
Says the person who didn't remember it.
Okay, I was just studying and I like kind of remembered it, okay? From Monday to Thursday, 0 review. At least I had this down and I switched A and B. So that's like
You
75%.
started with 6.
Okay, no, okay, so 6, no, but 6,
All
it
6
go,
is G0.
G0.
Yeah, no.
Yeah. It's just not included in this. So that's that. Let me go use the restroom. I still got like 15 more minutes of material.
Well, that's all I need to learn.
I'll be back right now. We're almost there. You look real tired, bro.
I'm writing little notes.
Yeah. I don't think so. No, we have no. Our test is in the morning.
Isn't it supposed to be the 12th?
It's Monday.
Next Monday.
Yes, it's 12. Oh, he might have. Yeah. No, he has his own file.
I just want
Ask.
no pressure. No pressure.
Did you ask?
Actually, this is our final PLF. Correct?
Yeah.
Okay.
I mean, that's what I think.
I assume, but
I don't
there
know.
was-
I will. Yeah. I'm pretty sure there's no PLF during finals week, right?
I don't think so.
I don't think so.
I mean, our finals also on said like that first Monday at
Oh,
10.
then yeah.
So,
Yeah. So. So there's two checkpoints, two main checkpoints. The first one is a G1S, which is gonna make sure that before we begin synthesis of DNA, that we do have good DNA. And then the second checkpoint is gonna be G2S, G2M, between G2 and mitosis. And that's gonna ensure that before we commit to mitosis, that we're good. So G, once again, stands for growth. So G1 and G2 are both G, growth 1, growth 2. Synthesis, S stands for synthesis. So we're synthesizing DNA. M stands for mitosis. Mitosis is prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. Then during mitosis itself, there is a checkpoint in the metaphase, which is the mitotic spindle checkpoint, which ensures that everything kinetochores are attached to the microtubules, and there is tension, and then we're gonna actually separate the chromosomes into two. And then there's also a checkpoint at cytokinesis, I believe, just like that. We did separate. So centromeres, centrioles, and centrosomes. Yes, centromeres are on the chromosome in the middle. Centrosomes are at each pole, and then centrioles are what makes up centrosomes. Okay. There are like the specific pathways, but we went over them specifically with you guys like in person. So, but I think you should spend some time on those when you're studying. Okay. Um, I don't know. I think that pretty much sums it up. Is there anything else you guys?
Recommend specific